My name is Danyelle Winchester. I'm the Common Funds Communication and Evaluation Liaison for the Kids First program. I wanted to share with everyone some impact the program had over the past 5 years. The program released 10 data sets representing a variety of pediatric conditions. You can learn more about these data sets on the Kids First DRC website, Kids First Umbrella Bio Project page, and the EXO1 abstract page on the Kids First program website. The value of the Kids First dataset will be amplified when researchers use and analyze these data to make discoveries that will ultimately improve prevention, diagnostics, and therapeutic interventions for these conditions. Researchers are using Kids First data to answer new scientific questions, as you've seen on some of the presentations thus far. Also, other scientists are using Kids First data to answer new scientific questions supported by NIH funding mechanisms, such as the Kids First RO3. From 2016 to 2019, 13 small awards were funded for analyses of Kids First data. 2 R1s were awarded as well. The Kids First program spurred new collaborations with other NIH initiatives, such as COMP2 and Include. Scientists are using Kids First data to provide insight into neuroblastoma, arthrogryposis multiplex congenita, or facial cleft, which you have heard from Dr. Marizita's team, and congenital diaphragmatic hernia. Over 2017 to 2019, a total of 13 papers were published from the Kids First program. RCR, which stands for Relative Citation Ratio, is a citation-based measure of scientific influence of a publication. The average RCR score for Kids First publications, which is highlighted in the green box, equals 1.32. This indicates that on average, Kids First papers have the same influence as an average NIH-funded paper in its field. The findings from the papers outlined in red were presented during a Kids First webinar. All manuscripts that include the use of Kids First data must acknowledge the data sets or sets by listing the dbGaP accession numbers. The acknowledgement statement for Kids First data can be found at the bottom of the dbGaP study page in the data use certification. I provided a screenshot of the statement at the bottom of this slide. We also covered how to acknowledge Kids First data in the frequently asked questions for EXO1 cohorts selected for sequencing question number 5, which is available on the Kids First website. A complete list of Kids First publications can be found on the Kids First website under the Publications Search page, which I've highlighted in yellow. In my next slide, here's a view of the Publication Search page. Next slide. The last thing I would like to share is you can watch past presentations from Kids First investigators on the Kids First DRC YouTube channel. The DRC posted presentations from Dr. Chung and Diskin. And now I can turn it back over to Valerie.
Thank you so much, Danyelle. So now I'll provide an update on our strategic planning. So in late 2018 and early 2019, we probed the community, our researchers, our internal NIH working group, and our external program consultants for feedback to help guide the future of Kids First. In this exercise, 7 key recommendation themes emerged, and while we're not ready to announce new initiatives for the last few years of the Kids First program, we're going to tell you a little bit about how we're continually finding ways to address this feedback. So one theme that emerged was the challenge and opportunity for identifying structural variants that may underlie childhood cancers and structural birth defects. Structural variants include large genomic aberrations, such as DNA sequences that are inserted, inverted, deleted, or duplicated within genomes, and you heard a little bit about these earlier today. But sometimes they're difficult to detect with standard short-read sequencing technologies. So one way to address this would be to implement long-read sequencing technologies for some of these cohorts. This year, we are offering long-read sequencing for EXO1 cohort projects as part of a pilot. Kids First sequencing centers offer a variety of long-read technologies, such as those from Pacific Biosciences and Oxford Nanopore. And the goal of this pilot is to leverage these approaches to uncover genetic variation that may have been missed with short-read sequencing or to further inform findings from previous sequencing. So this is an expansion of the cohort sequencing initiative. Another challenge that has become increasingly apparent is the need and opportunity for additional clinical and phenotypic data efforts, such as data extraction, curation, harmonization, and integration to make Kids First data sets stronger and to facilitate the ability to identify similarities and differences across cohorts. The Data Resource Center and partners from the Monarch Initiative recently hosted a webinar to demonstrate the importance of deep phenotyping and to brainstorm ways to improve phenotype data collection, harmonization, and genotype-phenotype analyses. The webinar featured presentations from Kids First investigators who shared details on their phenotyping efforts. Overall, the presentations highlighted the richness and complexities of Kids First phenotypic data, as well as opportunities for integrating diverse data and tools with the Kids First data resource to further empower cross-disease, cross-data type, and even cross-species discovery. As you saw today, phenotype harmonization and the new ontology browser in the portal enable us to view and study the intersection of phenotypes across various data sets. So the more phenotyping efforts we pursue, the more useful the data resource will be to empower new research questions. Given the momentum around this effort, the Data Resource Center intends to host a part 2 of the phenotype webinar later this summer. Another important recommendation that is really core to the goals of Kids First is to enable researchers to cross-analyze Kids First data with other pediatric data. The Kids First Data Resource Center is part of a new NIH cloud-based platforms interoperability effort, which is a collaboration with NHGRI Anvil, NHLBI Biodata Catalyst, and the NCI Cancer Research Data Commons. The goal is to enable and promote end-user analyses of human genomic and phenotypic data sets across these platforms through federation and interoperability. As you've seen during previous Kids First data resource demonstrations, the data resource already has some interoperability with NCI's pediatric cancer from the Target program. And this new interoperability effort is being driven by additional key use cases that our research community, you all, have identified. For example, the desire to cross-analyze data from the Pediatric Cardiac Genomics Consortium that has been sequenced by both TopMed and Kids First. Like the Kids First data resource, each of these platforms are made up of portals for finding and aggregating data, cloud-based analysis workspaces like Cavatica, collections of analytical workflows and tools, and cloud-based data storage repositories. And the goal is to establish guidelines and technical standards that will empower end-user analyses across these platforms. Over the last 6 months, the group has established working groups to address various layers of interoperability, including addressing operational barriers to trans-platform data sharing, testing technical standards for exchanging data across platforms, coordinating training resources for transitioning researchers to the cloud, and piloting FHIR standards for modeling and exchanging clinical and phenotypic data. The groups are making tremendous progress on these critical first steps to achieving interoperability, but additional activities will, of course, be needed. Currently, all of these platforms use different methods of logging in and managing approvals to controlled access data, and this is one of the major roadblocks to interoperability. The NIH CIT is therefore working to create one unified research auth service, or RAS, that will streamline access to multiple data resources. You saw today how many systems you have to log into just within the Kids First environment to integrate and access data across various sources, so RAS will even simplify Kids First data access procedures too. This group is making good progress, and they plan to deploy components of the service for systems that enable single sign-on and modernize communication of dbGaP approval information this summer. As we've discussed during previous webinars, we are continuing our collaborations with the Include project and the Knockout Mouse Phenotyping project, as well as beginning to engage in emerging opportunities with the Common Fund data ecosystem. And through these collaborations, we hope to continue addressing the recommendations and the needs of the research community. Now, I know we've been answering questions in the Q&A throughout the webinar, so I'm going to go ahead and jump to answering some of the questions we haven't answered yet, but were very common in the registration system. So the first question is what funding opportunities are available. We have an FAQ on our website that summarizes this entirely. But as of right now, the only funding opportunity that we plan to reissue that uses Kids First funds would be a reissue of the Kids First cohort sequencing opportunity. Using funds from various ICs, you can apply to the Kids First RO3 PAR for data analysis and data tool development, as well as the Parent RO1, and there's also a Parent RO3. To validate variance with model systems, we've listed some FOAs that might be relevant to you. And although we're not accepting proposals for our collaboration with Comp2 right now, you can still contact this email address to pursue additional collaborations. How can I get involved is also a common question we received. You're in, welcome to Kids First. Please go ahead and log into the portal and search across the data and play with some of the tools that are available in the portal to everyone. You can also contact the NIH mailbox or the Data Resource Center mailbox for feedback, and you can visit these websites to learn more. Another common question is, how do I access data? Like we've talked about, anyone can log into the portal to search across data, looking at de-identified metadata. But if you are going to want to access individual level sequence data, you will have to submit a dbGaP data access request in line with the NIH genomic data sharing policy. Once you have access, you can check out these resources for pushing the data you have approved access to in Kavatica. And during previous webinars, we've talked more about dbGaP data access requests, and so you can watch that previous clip or visit this link as well. And we received a question about how sequence data are released through the data resource. David, do you want to answer this one?
Question that a couple of our users asked was, how are sequences released? And so we collect data from two different groups. So our XO1 research groups provide patient phenotypic data, and as well as we receive the sequencing data from our sequencing centers. And so we at the Kids First DRC, we do what's called harmonize that data, and so get all those different lines of code to line up perfectly with each other, kind of double check everything, and then we release that to the broader community. And so for each sample, we release the complete genome sequence, and that's in CRAM format, as well as we do some initial steps of analysis. We release the unique differences in both VCF and GVCF formats.
We also received several questions about how community researchers can start connecting with each other. Adam, did you want to talk about this?
Sure. As I highlighted, briefly, we're really in that phase where, as you saw in all the presentation, really need to bring people together, not just within laboratories, but across the community. We've made early efforts on this within the portal. So for example, there's a new feature called the Kids First membership, where you can choose to make your portfolio public so that people can find you, connect with you around your interests and activities. Also, you can always share and publicize any of your activities via a variety of different environments. And this is true in almost every single one of our platforms. I saw a question in the chat asking about public and private projects. Yes, you can make your project public as long as they are meant to be public, and also engage in public projects that are already in Kavatica. But likewise manage and bring in members of the community into your own membership space, as long as they have the correct controlled access requirements that Kavatica will check.
Thank you, Adam. We have just 1 minute left, and I think there's just a final question about, is there source code for the data portal available?
So I can answer that. Yes, everything is public, available, open source. You can find the code on the GitHub repository, the Kids First repository.
Yeah, we can make sure we share that link with you, Andrew. Well, we're pretty much at 5 o'clock, but I will wait until 5 o'clock to see if any more questions come in. Okay, it is 5 o'clock. I do apologize that we didn't have as much time for questions as we wanted, but I think we were able to answer questions in real time using the Q&A. We received a few really interesting questions that we plan to follow up with some of you individually by email. And all of this will be recorded and shared eventually. So please be on the lookout and stay in touch and sign up for the listserv. Thank you all so much.
Thanks, everyone. And thanks to all the presenters.